Welcome everyone, and you're listening to Neutralizing COVID, the monoclonal antibody story, the podcast where we unpack the science, strategy, and real world impact of monoclonal antibodies in the fight against COVID-19. I'm your host, Dr. Shikha Agrawal, and today's story is about the race to turn lab discoveries into life saving hospital treatments. Join me for From Bench to Bedside, how monoclonal antibodies were rapidly deployed in a pandemic. Back with us is Dr. Aiswarya Chaudhuri, a nanomedicine expertise with a focus on targeted drug delivery, who will help us make sense of the science behind these themes. Welcome to the show, Dr. Aishwarya.
It's wonderful to be back, Dr. Shikha. I am excited for today's topic.
Let's start from the beginning. Before COVID 19, monoclonal antibodies were used for cancer, autoimmune diseases, even Ebola. But most people hadn't heard of them until 2020. What changed?
What changed was her urgency. Now, SARS-CoV-2 was spreading globally and fast. Vaccines take time to roll out, and small molecule antivirals weren't ready. So, monoclonal antibody became one of the most promising tools we had for immediate intervention. Now, scientists already knew how to make them. We just had to adapt them for this new virus.
Speaking of that, how did scientists identify which antibodies could work?
Great question, Dr. Shikha. In the first months, researchers screened blood from people who had recovered from the COVID-19. They isolated the B cells that were producing the neutralizing antibodies, ones that could actually block the virus from entering the human cells. Then those antibody genes were cloned and mass produced in labs.
That sounds like a long process. How did they make it happen so fast?
It normally takes years, but several things speed it up. First, we already had the platforms for making the antibodies. We weren't starting from the scratch. And second, the genetic sequence of the SARS-CoV-2 was published publicly just days after it was identified. That led the labs all over the world start designing the antibodies right away.
So we had speed, platforms, and collaboration. But deploying a treatment is a whole other step. How did regulators respond?
That was historic. Emergency use authorizations, EUAs, were granted for the first COVID-specific monoclonal antibody in the late 2020. These were based on strong preclinical data and early clinical trials, showing they could reduce viral load and prevent the hospitalization when given early.
So what about manufacturing? Were there enough doses for the people who needed them?
Initially, no. Manufacturing monoclonal antibodies at scale is complex. You are talking about living cells producing proteins in bioreactors. The US and other governments partnered with companies to wrap up the production fast, but demand still outweighs supply for a while.
Did these antibodies stay effective over time?
That's where it got tricky. SARS-CoV-2 mutates. So some variants developed spike protein changes that made certain antibodies less effective. These led to the withdrawal of some EUA products, but researchers kept testing and outdating the antibody cocktails to match the circulating trends.
So it was a constant cycle of adaptation.
Exactly. Surveillance, lab testing, and regulatory responsiveness all had to work in sync. It's an example of how pandemic science requires flexibility or not just speed.
Let's talk about impact. What role did these antibodies play in the bigger COVID picture?
They helped bridge a gap. Before vaccines were widely available, they were one of the few targeted treatments we had. For high-risk patients, especially early in infection, antibody infusions reduce the chances of several illness. In some cases, they also helped protect people who were exposed but hadn't yet developed the symptoms.
Were they used for prevention too?
Yes, some monoclonal antibodies were authorized for post-exposure or even pre-exposure prophylaxis in high-risk individuals like immunocompromised patients who might not respond well to the vaccines.
Looking back now, what would you say the antibody rollout taught us?
It taught us that the scientific infrastructure matters. The ability to respond quickly didn't come out of nowhere. It has built on decades of research into viral immunology, antibody engineering, and biotech manufacturing. It also showed that the rapid translation from research to clinical use is possible when the systems are aligned.
Before we wrap up, anything you want listeners to take away from this?
Yes, and monoclonal antibodies weren't a silver bullet, but they were a critical part of the pandemic toolkit. They showed what's possible when science, industry, and government work together under pressure.
Thank you, Dr. Aishwarya. This was a clear and insightful look at how monoclonal antibodies went from lab discovery to the frontline defense.
Thanks for having me.
That's it for this episode of Neutralizing COVID, the monoclonal antibody story. If you're enjoying the series, follow the show and share it with someone curious about the science behind pandemic solutions. Until next time, take care and keep exploring science.